Welcome back to our third lecture on fluorescence sensing. During this lecture, we're going to talk about chloride sensing and the use of yellow fluorescent protein for sensors. Chloride, I decided to keep as a separate topic because all of the principles and the reasons are different for chloride. Chloride could vary over a wide range of concentrations. Chloride does not fit in with the categories of the other probes, like the ion probes, where they have a distinct chelator for the ion of interest. So different sensor strategies were needed. Just in terms of importance, I looked up some medical reasons. Chloride is elevated in airways and cystic fibrosis. I remember reading this paper a long time ago where the probes were just simply put into different parts of the lung tissue. Interestingly, chloride is an initial signaling event for apoptosis, or programmed cell death. I looked at this paper carefully because I thought they must mean it's a consequence of apoptosis, but no, they indicated it's one of the signaling events that starts apoptosis. Chloride is different in different organs, so we couldn't just use one pH probe or one sodium probe, because there'll be a very different chloride concentration in different parts of the body. Even in cells, it can be different. In neuronal cells, it's different in the dendrites than in the cell bodies themselves. Low chloride can arrest cell development, and low chloride can affect tubulin polymerization. So the measurements are needed over a wide range of conditions and a wide range of concentrations. Let's look at the probes that we have available for chloride. The probes we have available are all quinolines or acridiniums. There's only one acridinium here. They're all electron deficient species, and you can see they all have very different quenching constants, as seen in the Stern-Volmer plots. This is valuable because the chloride probe can be selected for whatever chloride concentration range you would like to work out. Of course, the luginescens or lucigenins have a different, longer absorption spectra because they're a bis acridinium complex. I put those two absorption spectra here just to give you an idea why lucigenin is a different type of probe. The SPQ type probes, they just have unstructured absorption and emission. This bis-acridinium type probe, first of all, it has a longer excitation wavelength, meaning they can be used in cells with less exposure to UV, and of course the emission spectrum is also at a longer wavelength. The mechanism of quenching, it's most likely a PET quenching mechanism, photo-induced electron transport. You can tell that by looking at the quenching constants for a variety of different ions, and you see that the quenching constants increase for ions that are most easily give up their electrons. So iodide is more effective than chloride because iodide is more willing to give up an electron in a PET process. There are problems with chloride sensors. Most of them require UV excitation. Cells don't always like that. Could damage the cells, get a lot of autofluorescence at shorter wavelengths. The chloride probes don't display a spectral change, as opposed to the pH sensors and the calcium sensors we looked at, they cannot be used as wavelength ratiometric sensors without some further modification. The chloride sensors tend to leak from cells so that the intensity becomes unreliable. And for that reason, lifetime-based sensing is commonly used with chloride probes. And I guess a difficulty to using them is I looked at a number of catalogs, and conjugatable versions or AM versions of these probes don't seem to be available. So they're hard to use because you can only let them diffuse into cells and they can diffuse out. So here's an example of using lifetime imaging of one of those probes seen on the previous slide. The probe contained this probe, and measurements were made at different points in the cell. And when they measured right on the dendritic knobs, which are on the end of this part of the nerve cell, they got a shorter lifetime. When they looked at the supporting cell body, it was a longer lifetime. No information on the chloride concentration is available from the just the intensity images, but here the lifetime images, and we'll talk about lifetime imaging in a future lecture, but the lifetime imaging could be converted into chloride concentrations, and the lifetime indicated that the chloride concentrations were much higher in the dendritic knobs than they were in other parts of the cell body. So you can localize the probes. Well, you can, if you measure fast enough, it's not going to leak out, and you can get the measurements done. This is sort of a historical point, but that I found interesting, and I'll just take you back to the first lecture that we had, where we spoke about Stokes and Stokes discovering the Stokes shift. And I found some papers that claim that Stokes also observed quenching by sodium chloride of quinine. Quinine looks like many of the chloride probes. This implied that Stokes discovered chloride quenching of chloride. I don't know if that's true or not. I can believe he put it in the NaCl. I don't know how he decided which ion quenched, but several papers say this, but I could not find any of these older papers. But there's no doubt that chloride quenches quinine, and there's a good chance that even Stokes observed this at some point in the past. Because the chlorides probe leaked out of cells, what was needed was a conjugatable form. One or two groups did make a conjugatable form. They made it out of the leucinogen because it was most favorable, longest wavelength, and they could make it in a reactive form. Excuse me. And the more interesting part of this experiment is they also attached the same probe to dextran that was labeled with tetramethylrhodamine. So now they had in one probe a chloride-sensitive probe linked to dextran that had a chloride-insensitive probe. And here we can look at the emission spectra, and we can see if we look at the TMR dextran itself, it's not affected by the chloride concentration, but the probe, the chloride-sensitive part was very sensitive to chloride, and you could do the chloride concentrations from the wavelength ratiometric measurements. Importantly, the dextran didn't leak out of the cells, so that helped prolong the experiment. And as we'll see in a little bit, the quenching didn't depend on pH. That might not seem important now, but it will in a few more minutes. Problems with this approach is the probe uptake can be slow if you want the cells to take up dextran, and dextran, which is conjugated to a couple of fluorophores. It can take many hours or 24 hours, and so the cells are often labeled by microinjection that complicates the experiment. Then we had a remarkable discovery, which changed how chloride concentration measurements would be done in the future. While looking for other chlorides, other color GFPs, they made a yellow fluorescent protein, and lo and behold, they discovered that the yellow fluorescent protein was sensitive to chloride. That was not an aim of the original experiments, but it just turned out to be true. And this happened with this genetically encoded yellow fluorescent protein. The big problem was with the pH was that the quenching was pH dependent. So let's say you had a fluorescence measurement here, and you wanted to know what the chloride concentration was. If you exactly knew the pH inside the cell, you could then figure out what the chloride concentration was. But you usually don't know the exact pH inside a cell, or if you don't know the exact pH where the probe is located. So this very much restricted the use of this chloride-sensitive GFP. One thing that was interesting about the GFP, the yellow fluorescent protein, is that the absorption spectrum changed. It was meaning this was a ground state change in the chromophore. So somehow the binding of chloride changed the ion distribution in the chromophore and the GFP, but it was only seen, it could be seen in the absorption spectra, which meant it was a ground state in exchange, and we consider that favorable because excited state processes are difficult to control. From my reading, preparing this lecture, I found out that chloride is increasingly important in brain chemistry research, and it's probably going to have a big effect in that area. Chloride is important in brain cells. It affects synaptic transmission. A high or low chloride concentration around the axon can affect whether the nerve impulse continues. It's responsible for memory, advanced processing of information, and routing of information because it's controlling where the nerve impulses go. One possible problem is that the nerve impulses are relatively fast. So what's going to be important is that these probes respond very rapidly to changes in chloride concentration. Now, in the case of the other quinolinium probes, it was obviously that they changed instantly with chloride concentration. That's not obvious at all with these probes, and some of them, the exchange rates might be too slow to be used to follow nerve impulses. One of the uses is trying to watch changes in chloride concentration along the axon as the nerve impulse travels down the axon. If you can get these probes to work, there are many advantages to YFP-based chloride probes. They can target specific, well, in order to do it, you have to, of course, put the gene into the cell. That makes the experiment more complicated. But because you have to put the gene into the cell, you can target different cell types Naturally, it's restrained within the cells after it's synthesized. It's more photostable. It's said to be independent of concentration. Some researchers claim that the quinoline-type probes depend on concentration because they bind to other macromolecules. That's not going to be the case with GFP because it's buried in that beta barrel, which is going to protect it from contact with other macromolecules. Unfortunately, the attempts to eliminate the pH dependence of YFP have only been partially successful. There's been many attempts to do this. This is the pH dependence of the first GFP shown in a different way, but you can see if you don't know the pH, you don't know the chloride concentration, and conversely. There's one mutant of yellow fluorescent protein that was made that at least shifted the pH dependent part out of the normal physiological range. So once you get above pH 7 or so, its response is independent of pH. But this solution was not general enough, so you couldn't transfer it to other tissues. And some other solution was desired to make the YFPs independent of pH. So the concept that came up was to use dimers of GFP, just like inserting GFP into cells. You can insert longer gene sequences that contain 2 GFPs. And the initial thought was, well, you would use one GFP that was sensitive to chloride, another GFP that was not sensitive to chloride, and you would link them together. I think the drawing like this was done because it was reasoned that binding of chloride would change the amount of energy transfer between the two different colored GFPs. These dimers are called, here we go with trying to pronounce the word again, chlomylions, which was a play on the word of chameleons, which do change color. These seem to work quite well. You could, when you hook the two together, or if you looked at one alone, you would see that the YFP version would be sensitive to chloride. The other part of the dimer would not be, and of course that results in a shift in the ratio of the emission intensities at the two wavelengths. So you can use wavelength ratio measurements to determine the chloride concentrations. This is a difficult plot to understand completely because of the unusual the way it was normalized. But anyway, it shows that the ratio of the emission from the two chloride, the one chloride and insensitive protein will change in response to chloride concentration. It's not clear to me whether the changes in response to chloride are due to fret or not. In fact, you can find different graphs in the literature, some just listing the wavelength ratio, some listing the fret ratio. In my opinion, some fret occurs. I'm not sure that it's the complete reason for the chloride sensitivity of these dimeric proteins, but in any event, they're going to be widely useful in brain research. Then another miracle occurred. Someone asked the question, can a yellow fluorescent protein be made that displays enhanced fluorescence instead of quenching? I can just imagine what happened in the NIH study sections when this was proposed. All the reviewers would know about the quenching of the protein, and trying to look for one that was enhanced was probably deemed to be impossible. But in any event, the scientists succeeded in doing this by using high throughput screening. So basically, they started with a parent GFP. For some reasons, they made a few modifications before they went on to making random genetic modifications. So then they round up with hundreds, or if not more, versions of this parent GFP, and they looked for ones that showed the best response to chloride. They called that a chloride-orn probe. So they set a threshold around 4, and numerous mutants were tested, and they found that the certain ones displayed greater than a fourfold increase in intensity in response to chloride. So that's the exact opposite of what we've seen so far with the chloride quenching of yellow fluorescent protein. And the results, I thought, were pretty remarkable. I saw no reason to separate these, but they came up with at least three of these GFP-type proteins that displayed enhanced fluorescence in the presence of chloride. So this was wonderful. First time it's been done. And some of them display especially this number 1, very small change, no change at all in the absorption spectra. So there's no change in the ground state, a confirmation. So this is, I always like a probe that displays an enhanced fluorescence rather than being quenched. So these, I think, will become widely used in brain chemistry. And they were insensitive to pH. I was bringing up this pH problem all through this lecture, but these particular probes that turned on with chlorine, they didn't have any significant pH sensitivity in the physiological range. Here they rapidly changed the pH and just different concentrations of chloride were added, and they all pretty much behave the same. So based on this recent progress, I would believe will be extensive use of these GFPs in brain research. So that's the end of the sensing lectures, the end of 11 Part C. I hope breaking these lectures up made the whole topic more understandable to you. Thank you for your attention. Bye.